Gentlemen, you're all set up?
Yeah.
Yeah. Yep, I'm ready. Remember.
Now you may be wondering, why is this music playing? Well, this is the main music from Jurassic Park. This movie has heavy ties to cloning. If you watched the movie before, you may not realize, but it does. Our group actually got our research question from a quote from this movie. The quote is, "Life finds a way. God creates dinosaurs, God kills dinosaurs. God creates man, man kills God. Man brings back dinosaurs, and scientists were so preoccupied with whether or not they could that they didn't stop to think if they should after being out there." The research question that we got from this was, is preserving life through cloning the best route? And the best route is standing for the best route for humanity. Will it help preserve our life? To start our group, Ken and Case will be looking at the benefits of cloning through the scientific lens.
Thank you, Camden. I am now going to dive into the scientific lens of cloning. In the 1880s, Hans Dijkstra showed the earliest demonstrations of cloning involved artificial embryo twinning. He showed that sea urchin embryos could be split and developed into two separate individuals. This is when the question comes around, is preserving life through cloning the best route for humanity? I will explain scientifically why cloning is the best route. First, I will explain what exactly cloning is and what it can be used for. Then, I will show the science and technology behind cloning. Following that, I will explain what therapeutic cloning is and how it functions, and finally, I will define and explain what de-extinction is and how it can be used for our society. To start my speech, I will explain what cloning is and its multiple uses. The National Human Genome Research Institute defines cloning as it relates to genetics and genomics as involving using scientific methods to make identical or virtually identical copies of an organism's cell or DNA sequence. The phrase molecular cloning typically refers to isolating and copying a particular DNA segment of interest for further study. Many of you think of cloning as just making an exact copy of someone or something. Yet that is not wrong. There are many different ways or reasons why you can clone something. There are three main types. The first type is gene cloning, which is a common practice in molecular biology labs that is used by researchers to create copies of a particular gene for downstream applications such as sequencing, mutagenesis, genotyping, and heterogenesis, expression of a protein. This is a technique commonly used today for isolating long and unstudied genes in protein expression, as stated by BioLab. The second type of cloning is reproductive cloning, which is defined as the deliberate production of genetically identical individuals. Each newly produced individual is a new clone of the original. Identical twins are natural clones. Clones contain identical sets of genetic material and nucleus, the compartment that contains the chromosomes of every cell in their bodies. Thus, the cells from two clones have the same DNA and the same genes on the nuclei, according to the National Library of Medicine. And finally, the third type of cloning, which I will be covering more later on in this speech, therapeutic cloning. Therapeutic cloning involves creating a cloned embryo from the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These stem cells can be used in experiments aimed at understanding disease and developing new treatments for disease, stated to the National Human Genome Research Institute. The idea of human cloning was science fiction when it was first imagined, but in the last few decades, technological and scientific advances have made this a real possibility. Although the ethics of cloning a human are questionable, the technology has led to promising reproductive and health therapies. As cells grow and divide, they naturally create clones using cellular division, a process called mitosis. The cells use proteins and enzymes coded in their genes to copy their genetic material. As researchers developed an understanding of how cells reproduce their genes, scientists began recreating these reactions in the lab. Now, cloning genes in the lab is as easy as mixing a drink, combining the proteins that the cells use to copy their DNA and adding a gene to copy. And I bet most of you are asking yourself the same question. Have humans ever been cloned? Sadly, no, but there's a type of cloning that can help the human body function better than before. And perhaps the most medically applicable type of cloning for humans is therapeutic cloning, which creates cloned embryonic stem cells of a patient to create genetically identical cells that can treat a medical condition. Therapeutic cloning refers to the use of embryonic stem cells that in our lab can derive from somatic cells from a patient's skin. That is from an embryologist at Oregon Health and Science University in Portland. Researchers hope to use embryonic stem cells, which have the unique ability to generate virtually all types of cells in the organism, to grow healthy tissues in the laboratory that can be used to replace injured or diseased tissues. In addition, it may be possible to learn more about the molecular causes of a disease by studying embryonic stem lines from cloned embryos derived from the cells of animals or humans with different diseases. Finally, differentiated tissues derived from each cells are excellent tools to test therapeutic drugs. Many researchers think it is worthwhile to explore the use of embryonic stem cells as a path for treating human diseases. However, some experts are concerned about the striking similarities between the stem cells and cancer cells. Both types can proliferate indefinitely, and some studies show that after 60 cycles of cell division, stem cells can accumulate mutations that can lead to cancer. Therefore, the relationship between stem cells and cancer cells needs to be more clearly understood if stem cells are to be used to treat human disease. Now you have the understanding of what therapeutic cloning is, I will shift my topic over to explaining what de-extinction is and how it can be useful. De-extinction is the process of resurrecting species that have died out or gone extinct. Although once considered in imagination, the possibility of bringing extinction species back to life has been raised by advances in selective breeding, genetics, and reproductive cloning technologies. In the later part of the 20th century, tools emerged that enabled scientists to isolate and analyze DNA from bones, hair, and other tissues of dead animals. Coupled with advances in reproductive technologies such as in vitro fertilization, researchers were able to identify cattle that are close genetic relatives of the aurochs and combine their sperm eggs to produce an animal that is morphology and genetically similar to aurochs. De-extinction provides an opportunity for humans to rectify the past harms inflicted on other species, as well as to expand species diversity. But many extinct species were driven out of extinctions as a result of habitat loss, and others live in habitats that have since been altered dramatically. In addition, in the near term, resurrected species would be considered endangered and would therefore require conservation, for which brings resources often constrained or lacked. De-extinction, by providing the option to bring species back later, also could have the unintended consequence of condoning extinction and could have future endeavors in threaten biodiversity. Now that I have explained my main points, it's time to give you an overall conclusion of this speech. In this speech, I started by explaining what cloning is, how it works, and giving you a detailed definition of the term. I then transitioned over to my second body where I explained 3 different types of cloning and thoroughly explained therapeutic cloning. I then gave you my last body explaining what de-extinction is and how it could affect our society, and what science could lead us in the future. Though the scientific lens shows how cloning is beneficial for humanity, Hallie will now show you guys the historic lens and how it could be a different perspective on how cloning is the best route for humanity.
Thank you, Cannon. As Cannon said, I will be informing you on the historical background and hopefully give you another perspective. Have you ever wanted to be in more than one place at one time? Well, I can assure you, many other people have had the same thought, specifically scientists. Samuel H. Wood, a fertility specialist, attempted this idea in 2008 when he created 5 mature human embryos with his own DNA. This experiment was just a small glimpse into the future of cloning. So today, I'm going into depth about the history of cloning and how it has shaped the generation of work that we have today, as well as the different kinds of cloning. This journey through time not only reveals the scientific advancements that shaped our understanding of replication, but also how we got here. To start, cloning in simple definition is to create a genetically identical copy of an organism. This concept has roots that stretch nearly back to ancient times, when nature itself displayed cloning through methods like asexual production in plants and the occurrence of identical twins. However, it wasn't until the 20th century that scientific community began to understand the mechanisms behind cloning. So what kinds of cloning are there? According to Learn Genetics Utah, there are 3 types. First is basic embryo splitting. This is the mirroring of identical twins where the embryo splitting early in development. Next, we have nuclear transfer. This is where the nucleus from a donor with DNA is transferred to a nucleus removed egg cell. Then, finally, molecular cloning and beyond. This is where a gene is isolated and inserted into a vector to be replicated within its host organism. Now, since we have an understanding of what each type of cloning is, we can dive into the history that shaped cloning today. Let's begin with the first documented experiment of cloning, starting in 1885. Hans Adolf Edward Driesch was the first to have documented the cloning of a sea urchin. Driesch separated the embryos of the urchin, and then they grew into 2 separate sea urchins. Excuse me. This experiment was demonstrating how the body can grow from just a single cell and form a whole organism. Then later in the 1900s, researchers began experimenting with artificial embryo twinning, a technique that involved splitting early embryos to create identical offspring. This was done by Hans Spemann, a German embryologist who was Nobel Prize in Physiology or Medicine. He experimented with a salamander and discovered the cell nucleus controls embryonic development. After this, John Gurdon, another scientist but in biology, successfully cloned a frog, according to the Birth of Cloning by PubMed Central. These efforts laid the foundation for future advancements and researchers. As our understanding of genetics evolved, so did our ability to manipulate biological processes. The nuclear transfer of a differentiated cell in 1952 by James Watson and Francis Crick early on marked a turning point, provided a key to unlocking the secrets of hereditary and genetic replication. Now, if we fast forward to the 1970s, when scientists began to explore the potential of recombinant DNA technology, this breakthrough allowed for manipulation of genetic material, paving the way for advancements in genetic engineering. However, it was the 1996 cloning of Dolly the sheep that truly captured the world's attention. According to Frontline Genomics, Dolly was the first mammal to be cloned from an adult somatic cell. and her birth raised both excitement and ethical concerns. Her successful cloning proved that it was possible to create an exact genetic copy of a complex organism. Biotechnology opened the door to new possibilities in medicine, agriculture, and even discussions about the potential of cloning humans. Dolly's creation was a huge achievement that demonstrated the potential to replicate complex organisms. It opened new avenues for research in medicine and conservation. Scientists envisioned a future where cloning could lead to breakthroughs in organ transplantation and preservation of endangered species, and even the possibility of resurrecting extinct animals. Yet, alongside these possibilities came a wave of ethical questions. What does it mean to be able to do this? Should we clone humans? What are the implications for individually and genetic diversity? Following this breakthrough, researchers cloned other animals such as cows, pigs, goats, and even endangered species like the gar and the banteng, aiming to improve livestock breeding and conservation efforts. In 2000, advancements in the stem cell research led to the development of the induced pluripotent stem cells, offering a promising, less controversial alternative to embryonic stem cells. Cloning technology has also evolved with improved techniques, like somatic cell nuclear transfer, leading to higher successful rates and more ethical discussions around human cloning, therapeutic cloning, and potential applications for regenerative medicine. As years progressed, the debate surrounding cloning intensified. The scientific community grappled with the moral implications of their work, leading to calls for regulations and ethical guidelines. In some countries, human cloning remains banned, while others allow for the therapeutic cloning aimed at medical advancements. The differing perspectives reflect cultural values and ethical considerations that shape our understanding of life itself. To conclude what we have recapped in history, in conclusion, the journey of cloning, from earliest experiments with sea urchins to the groundbreaking creation of Dolly the sheep and beyond, has reshaped our understanding of biology, genetics, and what it means to replicate life. The development of different types of cloning, such as embryo splitting, nuclear transfer, and molecular cloning, has allowed scientists to explore the possibilities once considered science fiction. While these advancements hold great promise for medicine, agriculture, and conservation, they also raise important ethical questions about identity, individuality, and the boundaries of science. As we continue to innovate and push the limits of what cloning can achieve, it is crucial to balance scientific progress with thoughtful considerations of its impact on society and the world we live in. With all of this information, we can conclude cloning can be more beneficial to our world. Now, Camden will take a look into the ethical and legal side of cloning.
Thank you, Alec. So, what is ethics? According to Oxford Dictionary, ethics are moral principles that govern a person's behavior or conduct of an activity. Today, I'll be leading you through the information that I have discovered regarding the ethical view of cloning. I have done research on two topics specifically: religion and regulations. The reason that I chose those two to research is because they're the two biggest factors, in my opinion, on ethics. Religion is where most moral standards can be found through teachings and practices, and laws and regulations are just forced morals if you think about it. To start, we'll dive into the religious side of my research. I chose to research three religions: Christianity, Islam, and Buddhism. To start, let's take a look at the teachings of Christianity. According to a survey by Bob Sullivan, a technology consultant for MSNBC, 1 in every 3 Christians opposes cloning for religious beliefs. What are these religious beliefs that they're talking about? According to the Christian Institute, there are 4 main reasons, but I boil them down to 2. The 2 main points are God is the creator of life, and it should stay that way. And the other is that once human life is created, it should not be tampered with. As a Christian myself, I can testify that these are also the things that I've been taught as a kid. Many of us here know Christian views on most things because the Bible is a widely spread book. So how about we take a look at other holy books that are taught? Let's go deeper into the religious side of this argument and look at the Quran, which is the holy book for Islam. In an article published by the International Journal of Fertility and Sterility, it is stated that cloning does not contradict the theological teachings and concepts of the Holy Quran and Shiite Muslims. So obviously, since the Quran doesn't have anything to say about cloning, then Muslims must accept cloning as a moral and ethically sound practice. Well, that is not quite clear, because the Quran was not written in a time where cloning was even thought of. So how could you come up with an answer for something that wasn't even a problem back then? You just can't. But Dr. Abdulaziz Saqadini, a professor of the University of Virginia, argues that Muslim consensus on cloning will eventually emerge, suggesting that because cloning occurs, it must be permitted by God, or Allah. So he answers that since cloning is happening, then it is okay because Allah is permitting it. The International Journal of Fertility and Sterility supports the idea that cloning does not grant life, rather it utilizes the life bestowed by God. The hard part to find an answer with Islam, to make a decision, is that cloning technically does not go against their teachings because they don't have anything written about it. But many believe in the Islamic culture that someone who is cloned or is practicing that was sent by the devil himself. So Christianity has a clear view on cloning. Islam's is hard to find because it's just not talked about. So let's take a look at Buddhism. What do they have to offer? Buddhism is the only non-theistic religion that I chose, and it also has no holy book. So it doesn't have any book that they go to for teachings. According to the University of Hawaii, Buddhism might be willing to accept cloning, Campbell said, because it represents a leap in modern science and self-understanding that could only be considered a path to enlightenment. On the other hand, the Eightfold Path prohibits harm to any sentient being. Much like Islam, it is difficult to pin down. It's difficult to pin down. So we've looked at 3 different religions. Christianity had a clear view, but if you think about it, it kind of does go against their teachings because Christians are taught to love everybody, but if they met somebody that was cloned, they probably wouldn't. Muslims, you don't really know where they stand because they don't have teachings on it, but if it's in Allah's will, then they're permitting it. Buddhists don't have a clear view either. I didn't mention this, but they believe that someone that is cloned is devaluing themselves, which goes against the Eightfold Path, which is what they are taught. So, religion doesn't offer a clear view on the ethical side of cloning. So, where can we turn to when religion, which has strong moral principles, doesn't have an answer? Well, I chose to turn to laws and regulations because they are what the world has decided is the line, where you can't cross, where you should stay. For this, we'll be looking at 2 different points of view. We'll be looking at the legal side within the United States and also around the world. What does it look like for the regulations around the world? In the United States, according to Ruth Sorrell, there have been attempts to completely ban human cloning and even punish those who perform the acts of human cloning. Human cloning has not been banned within the United States, just regulated. I think the reason for this is summed up perfectly by Dr. Michael P. Scott. You don't have to deal with ethical issues, he said. The safety ones are enough. This is where the United States still stands on this situation. They don't have anything banning human cloning, but they do have regulations for safety. So what does the rest of the world look like? In an article by Adele Langlois, it says, many countries have banned reproductive and or therapeutic cloning. This goes back to 1993, when the United Nations Educational, Scientific, and Cultural Organization decided to start making regulations banning and regulating cloning. From 2008 to 2011, they worked on deciding whether or not to ban cloning of the human genome. They ended up dropping the topic without answering the previous predicament, but then in 2014, the bioethical team finally revisited the problem. In the end, they agreed there needs to be a ban on cloning the human genome, but they did not end up doing that. Why would they spend 6 years trying to decide something just to leave it hanging in the air? Well, the reason is that they concluded that human reproductive cloning is a challenging subject. Establishing a global governance framework in this area may be possible. In the end, a declaration was put out by UNESCO, or the United Nations Environmental, Scientific, Cultural Organization, that calls on states to prohibit all forms of human cloning inasmuch as they are incapable with human dignity and the protection of human life. So in the end, they prohibited human cloning, but human cloning is still happening in forms like therapeutic cloning that Cannon mentioned. Because the declaration by UNESCO is considered too weak an instrument to either thwart rogue research or promote legitimate scientific endeavor. In conclusion, the world and religion decided that human cloning is morally wrong. UNESCO made a formal declaration to get rid of it. Religions have turned their back on cloning because it either gets too close to playing God, clones are from the devil himself, or it devalues the person who's getting cloned. I personally don't believe in cloning. I think it is morally and ethically wrong. So I have agreed with the information that I found. So why does human cloning still exist to this day? And probably for the foreseeable future. Because it might be ethically and morally wrong, but it is the best way to save humanity from itself. Casey will now take a deeper look into cloning and how it is the best for humanity with the futuristic lens.
Imagine this. The person you love the most has been diagnosed with liver cancer and is in desperate need of a transplant. But the waiting list is infinite and the chances are small. This is not a hypothetical scenario. This experience is what Camden's dad went through. While society fears cloning and its threats on individuality, the real dilemma is the 100,000 people praying for a donor that meets their qualifications. According to the National Institution of Health, 5,600 people die annually waiting for a transplant. This is equivalent to 17 people dying each day, and another person being added to the list every 9 minutes. However, through cloning, this devastating fact can significantly decrease and become a breakthrough for not only people in need for a transplant, but also on other medical drugs, and potentially save endangered species from vanishing. While this concept may seem too far in the future, the reality is right around the corner, and scientists are already studying this process. So today, I'll be elaborating on the futuristics and advantages of cloning by looking at potential disruptions, long-term consequences, and hypothetical scenarios. This will allow for a better understanding on what steps have to be taken within society for cloning to grow in a beneficial manner. To start, the list of disruptions standing between cloning and today's culture is very long. So we need to explore what cloning is and what needs to happen for it to work. Michael Rugnetta, the author of Cloning Ethical Controversies, says cloning is the process of generating a genetically identical copy of a cell or an organism. This process can occur naturally when cells replicate themselves, asexually without any alterations genetically, or through recombination. Organisms that are prokaryotes will, like bacteria, can perform this. But eukaryotic organisms, such as humans, go through a different process called mitosis and meiosis. Although, as time goes on, scientists in the biomedical field have experimented with copying DNA in individual cells. This is done through a process known as polymerase chain reaction. Polymerase chain reaction is a common technique used in basic biological research, but as it evolves, it gets more complicated and controversial because of the usage of embryos. So in order to reproduce a clone, the embryos have to be implanted into a uterus, either artificially or a real one, then develop into a fetus and grows and carries full term. This complex process has been done, but it's considered to have a very low survival and gestation and be born without any defects. Adding to this method is a similar process called somatic cell nuclear transfer, in which they remove the nucleus of a somatic cell from an organism and insert it into an egg cell that had its nucleus removed. The egg cell eventually divides into an identical copy of itself, creating a clone. Somatic cell nuclear transfer is how Ian Wilmut and his team of scientists cloned Dolly the sheep in 1996, as Hallie talked about earlier. This technological advancements to make cloning possible is growing dramatically and will have to become more consistent and reliable before being used in humans. The next barrier to overcome and disruption to cloning is to establish guidelines and regulations for society. This will ensure the best use of such a complex system. Without structure, it can be taken advantage of and turned into the dystopian society we all feared for. These serious considerations should be determined by scientists, lawmakers, and citizens of the public to get the overall opinions of everyone. They will need to address safety protocols, hereditary soundness, and moral values. It's important to keep in mind the intended goal of cloning, which is to use it for the greater good in medicine, agriculture, and science. So, by learning from past disruptions, it will help future struggles. This way, we can create a more positive future and maintain human dignity. The last disruption that I think should be taken into consideration is all the environmental factors that could impact the development and growth of cloning. The uniformity in cloning can make it more vulnerable for new diseases or environmental changes to occur, creating devastating effects on society, or vice versa. The environment that we have may be too harsh for cloning to maintain and grow good health. So, keeping all these disruptions in the forefront of research, we have to be observant on how we approach particular matters and carefully weigh the odds between positives and negatives. Now, upon looking at all the potential disruptions, it's time to look at long-term consequences. The stability of cloning is very unknown, and there are many risk factors involved. But, as I like to think of it, every day we get up and we get into a motor vehicle, such as the school building that you guys are all sitting in right now. We take risks every day, knowing that outcomes could be detrimental, but it's worth it, since we get to experience the beauty of life created for us. The same analogy goes for cloning. This could be the solution of the century and create much beauty. Research from the University of Utah's Learn Genetics program discusses the reason for cloning and how the use can be beneficial for a large number of people. The article discusses the use of cloning can allow us to genetically match tissue for transplants, improve the understanding of genetics in disease, and pick favorite traits in animals. These factors would change the future and leave lasting effects. So it's important that we look at each one individually. Beginning with how it can genetically match tissues for transplant, also known as in a larger scale called therapeutic cloning. Therapeutic cloning involves creating new stem cells that are identical to that person, because stem cells are what build and repair the body throughout life. So it would offer a more natural process, and eventually it can be used for whole organs. The body wouldn't reject the organ since it's identically genetic to that person. Then, looking at the process of gene editing, it allows scientists to help cure genetic disorders. In 2013, scientists from Oregon Health and Science University successfully created a human embryonic stem cell, using DNA from an 8-month-old that suffered from a rare genetic disease. So, with farther observation, the team could have a better understanding about the disease and many others. But, on a side note, cloning also provides lasting consequences in animal life, because we can pick favored traits in livestock, or open up the idea of cloning extinct or soon-to-be extinct animals. In an experiment in 2009, scientists had almost resurrected a wild mountain goat called the bucado, using goats as the surrogates. The clone didn't survive soon after birth, but it gave a new speculation for scientists to study. Then, according to TheGuardian.com, a more recent experiment done in 2015 consisted of gene editing by extracting and editing mammoth DNA, then using Asian elephants as surrogates to carry it. These efforts have provided much intel to what the future could look like in the medical and in the animal science. Now, last topic I want to mention is the hypothetical scenarios faced along with cloning. This includes answering what-if questions and seeing what the world would look like in 20 years due to tipping and turning points. By doing this, it will allow for a better insight on what daily life could look like as an average human. So to start, what if we reach a point where cloning is reliable? Well, this means there could be limitless supplies of organs used for transplants, and other procedures would be much safer. Overall, decreasing the number of deaths yearly from illness. In addition, what if cloning becomes more than just organs in animals? If this happens, society could become a dangerous place and lead to violating rights and dignity as an individual, which is why ensuring legal laws is important. Next, foreseeing the future and daily life 20 years from now because of the advancements in this technology. It seems that it could be challenging for social norms, since a child is born from 2 individuals, and a clone would only be identical to one parent, challenging the responsibility and the roles. Also, cloning could be a question of identity and constant comparison, which would impact the mental health of that person. So, since breaking all of this down, hopefully the understanding of the futuristic cloning has become more clear. I believe, after looking at all the potential disruptions, long-term consequences, and future scenarios, it comes down to having a bigger and better positive influence on the impact of our future. We saw that cloning is discouraging in advancement, then explored the lasting effects in different areas like medicine, demonstrating that it could be the solution for humans fighting for the life in a hospital bed. Then we applied cloning into hypothetical scenarios 20 years from now to have a better vision. Therefore, as technology advances, so does society, and new solutions are being created. We must act in a responsible manner to ensure the best outcome. So, just like Jim Carrey says, life opens up opportunities to you, and you can either take them or stand afraid of taking them. The choice is yours to make, but the future waits for no one. Thank you.
So to conclude, I'd like to remind you guys of our research question. Is preserving life through cloning the best route? Well, our group came to the conclusion that it is. You can look at the scientific lens and see that the processes that you have to go through. You can look at the historical lens to see how we've gotten to where we are today. You can look at the futuristic lens to see where we can go from what we know. And the answer that our group came up with is that it is the best. And we'd like to leave you guys with a question. Do you think that preserving life is the best option? You can make a choice, yes or no. Thank you.
Okay. Question time. Can you defend what you just said? Cannon. Give one specific way that you that you changed results because of someone else's viewpoint in the presentation, or their individual sections?
I can honestly say that I feel like all three of them have kind of changed my perspective on like the history of how cloning has evolved in scientific and technology, and then the future of kind of scientific technology of cloning, and then the ethics. I feel like all around it's kind of changed my viewpoint. But,
Okay. Hallie, your question. Having finished your project, what, if anything, do you consider to be a gap in your team's research? If addressed, would make you feel more confident about your guys' conclusion?
Maybe the concerns, if we address more of the concerns, it would give people a wider perspective on how to choose.
Okay. Hamden, describe an argument from one of your peers' individual reports that made you think differently about your team's solution or conclusion.
I would choose my own because it might not be my teammates, but the conclusion that we came up with was that yes, cloning is the best route for humanity, but personally, through the ethical lens, I did not find that. It was the opposite. So I would say my own lens.
Okay? And then Casey, your question. Overall listening. Describe how the content of your team's presentation was changed as a result of your group discussions.
That is a great question. And I can say that from the beginning when we were choosing a topic, we didn't really know much about cloning. I know for me personally, that wasn't my first initial thought of cloning. So thinking about the positive wasn't even really in my thought or anyone else's thoughts. But then after taking a look at all of this research and learning all of the just different details about it, I can say that we have learned so much and that we definitely are for cloning. And looking at it at the beginning, it wasn't really there.
Okay, thank you guys very much. Do you have any questions?